facts about induction and repression. So we're going to start off here with induction on the left side. 3 facts. The first one, induction is a process that turns on transcription of genes or a gene. The second one, that inducers are involved, and inducers are substances that act to induce transcription of genes. Our third fact is that there are inducible enzymes, and these enzymes are synthesized in the presence of inducers. On to repression. Now, it inhibits gene expression and decreases the synthesis of enzymes. It responds to the overabundance of end product in a metabolic pathway, and the repressor that's involved in repression blocks the RNA polymerase ability to start transcription. The Operon model of gene expression was created by Francis Jacob and Jacques Monod. Here I have the inducible operon. The operon consists of a regulatory gene, promoter, and operator, also structural genes. Now the promoter and the operator, this is called the control region. And again, we have our structural genes. Now combine the structural genes and the control region will give us an operon. E. coli is a great example of an inducible system. So how does it work? Well, here's an example. Now, if there's no lactose present in the system, then the repressor binds to the operator and transcription doesn't take place. So if there is lactose present in the system, then the repressor binds to the metabolite of a lactose instead of the operator. Then transcription can go forward. Next up is our repressible operon. We have our regulatory gene, our promoter, and our operon. We have our structural genes, and in the repressor operon, it has a few more genes, E, D, C, B, and A. So if you combine the structural genes with the control regions, which is still the promoter and the operator, we have our operon, specifically repressible operon. So how does this work? So we have our structural genes that are transcribed and translated until they are turned off or repressed. So when there is too much of a product, the product can bind to the repressor. So we have our end product plus the repressor, so they bind, which creates our active repressor protein. Now this active repressor protein then binds to the operator site, not allowing the RNA polymerase to transcribe. So there's no structural genes, no enzymes. Alright, it's the study time portion of the video. I have our 2 words, induction and repression. And like in true Rochelle style, I have my main word and I pick a letter or more from the main word and I connect it to the definition. So first I have induction and I picked T. Now induction turns on transcription of genes. So here I'm going to write turn. Well, turns on. Induction, T, turns on transcription was the second T of genes. On to repression. Now the letters that I picked are R, I, and O. So I'm going to start with I, and the word that I picked is inhibit. It inhibits gene expression and decreases synthesis of enzymes. So inhibits gene expression, decrease synthesis of enzymes. So the next letter that I chose, I'm going to go with is O for overabundance. Now here, it responds to the overabundance of the end product of the metabolic pathway. So we have repression. It inhibits the expression of genes and it decreases the synthesis of enzymes. O for overabundance. So it responds to the overabundance of the end product of a metabolic pathway. So the next and the last one is R, and that is RNA polymerase. Rna polymerase. So here, the repressor blocks the ability for the RNA polymerase to start transcription. All right, we're going to review this. So we have induction, and we pick the T for turns on. So it's a process that turns on transcription of genes or a gene. We have repression, so we pick the R for RNA polymerase. So what that is that the repressor blocks the ability for the RNA polymerase to start transcription. We have I for inhibit, so that gene expression is inhibited and there is a decrease in synthesis of enzymes. And then we have O for overabundance. It responds to the overabundance of an end product of a metabolic pathway. All right, we're going to remember the Operon models. So it's IPO, that will always stay the same. IPO, regulatory gene, the promoter, and the operator. RPO, regulatory gene, promoter, operator. Now for the inducible, let me get a pen. The inducible operon, it's smaller. The structural genes are fewer actually. So we have Z, Y, and A. Structural genes. The control region is always P and O. The inducible operon is the structural genes and the control region. The inducible operon consists of fewer genes, Z, Y, and A. For the repressible, there are more genes in a structural genes. We have E, D, C, B, A. And again, we have our regulatory genes, the promoter and the operator. The control region is P and O, and then we have our structural genes. You put those two together and we have our repressible operon. Now to remember specifically about the repressible operon and how it works is that there's an end product that binds to the repressor, which gives us the active repressor protein. Now the active repressor protein then binds to the operating site. Inducible operon, we have our example of the E. coli. So when there is no lactose in the system, then the repressor binds to the operating site. Now, if there is lactose in the system, then the repressor binds to the metabolite of the lactose, and it does not bind to the operator. Okay, this is the test your knowledge section of the video. I have induction and repression. I want you to give me all of the facts about these two words. And we have our Operon Models. And I want you to fill in the blank, all of these blanks. And then I want you to tell me which one is the inducible operon and which one is the repressible operon. Hey my rockers, thanks for studying with me. I really did enjoy the study session. But before you go, I want to let you know of a couple things. The first one, I really would love for you to look in the description box because I have a free study gift for you. 2, I have one-on-one and group study sessions. So if you find that my videos are helping you in microbiology, feel free to email me. My email is in the description box. It is therock.astrongfoundation@gmail.com, and I will let you know all about my microbiology study program. Now, it is designed to be interactive and repetitive so that you can remember the material so much better. So again, thanks for studying with me, and I will see you in the next video.